Well, it's fun to be here and hi to everybody. So this title is a little bit, you'll see what I mean by it. Quasispecies is something that causes many people to run away, but mainly what I'm talking about is the birth of viruses within infected cells. And the people who work in my lab are here. I'm going to mainly feature the work of Jennifer, Dorothy, and Jasmine Moshiri here. Okay, so as you know, we're all interested in antivirals, and one of the biggest problems of antivirals is obviously drug resistance. So for direct-acting antivirals, as you know, there are a couple different approaches. The most successful thus far has been combination therapy, in which, if you say that drug resistance arises at 1 × 10⁻⁴ genomes, this cell obviously doesn't have 10,000 genomes, but let's say it has this one drug-resistant genome in it. And that's there at 10⁻⁴. If there's another drug, it might be a drug-resistant one, might also be present at 10⁻⁴, and one that's doubly resistant is 10⁻⁸. So that's to decrease the frequency of drug-resistant variants, and it's been very successful. But as you know, it's often very expensive also and takes a long time. There's another idea that we all kind of use in our minds, that's the most popular one, is to create a fitness cost to drug resistance. That is, here we have this drug-resistant virus growing up in this cell. That it was just born in this cell with a lot of drug-susceptible viruses, and it's very small because it doesn't grow. That's why I drew it that way. And the point is if drug resistance, the mutations that cause it, exact such a fitness cost that the virus can't grow, then you won't have a lot of selection for drug resistance. And that's been true of a couple of different drugs. Like, I think that's why sofosbuvir is so very, very effective. The question is, how do we engineer fitness cost? How do we know which amino acids are going to be so deleterious to virus growth that they will never mutate to be drug resistant? And that's one of the things we've been trying to think about. Something my lab has been working on for a while now is a different paradigm. That is, most drugs work by creating loss of function, like they'll bind to a protease or polymerase and cause it just not to work. But what about a compound that would bind to a viral protein and create a dominant inhibitor of other viral proteins and of other viruses in the same cell? That's why some of these drug-susceptible genomes here have little skulls and crossbones next to them, because our goal is to identify targets that, when bound to drugs, will inhibit everybody in the cell, including the drug-resistant one as soon as it's born. So what are the paradigms for that? Oh, well, first of all, I want to say that we've been working on this as an academic lab, et cetera, and imagine my delight when asked to work with the ASAP folks who have this incredible drug discovery platform that's structure-based, and to be part of the cadre that helps to choose the targets. So we found that the biochemistry of a few targets is such that when bound to drugs, they become dominant inhibitors. And some of those are included in the targeting pipeline. Jesse Bloom is also involved, and you guys have heard him talk here. And they have a really elegant way to try to assess fitness costs systematically. One is, so that's kind of here. How do you assess fitness cost, and how do you go after it systematically? Well, one way is to systematically target the substrate envelope to invariant residues. So invariant residues, what does that mean? Those are the ones that aren't out in circulation, that is so-called conservation. But we miss a lot in that way. And so the other approach is to do deep mutational scanning, which to examine all mutations in a coding region, and to say which ones, under which circumstances can the drug survive. So that's what some of the people in Project One are doing. But I did want to highlight something that Jesse's done recently, because it's such a remarkable addition that might be useful to all of you. I mean, how do we identify unfit amino acids by evaluating conservation? The problem with that is there aren't that many sequences, and there are bottleneck and dominance effects that skew the population. But what Jesse and Richard Nair have done is take the possible exception of 6 million SARS-CoV-2 sequences and said, okay, what amino acids are tolerated and what aren't at all those positions. This has been compiled. If you look at their bioarchives paper that's currently under review elsewhere, there's an interactive, and one of the things that Project One is very proud of, and we're going to continue to do, is in this case, if you look at the interactive map on their website, what you see is every single protein in SARS-CoV-2. You click on it, and then you see the relative fitness of all what amino acids occur in nature out of the 6 million sequences. So you can help to evaluate the fitness cost of, for example, if your substrate envelope is at, amino acid 20, you can say, well, there's a lot of fitness there. Maybe I'll choose somewhere else. So please consult that resource, and we'll continue to post such things on our website for everybody to utilize. Okay, so how do you find a protein that, when drug bound, is going to inhibit every virus in the cell? So one of the paradigms we worked on right away and have published a lot about is freely assembling oligomers like capsids. So if you have a drug susceptible viruses in the cell, and there's a drug resistant one that arises, if you have, for example, a capsid modulator, and this drug resistant protein makes a capsid that doesn't bind to the modulator, too bad for it, because it's going to co-assemble with all the other capsids in the cell and make chimeric molecules, chimeric variants that are on average drug susceptible. That sounds good in theory, but we've actually tested it in many cases. Capsids do mix, and drug resistance is suppressed in the presence when capsid modulators are used. Today, I want to talk to you about a different paradigm that we've been working on lately, because the ASAP Consortium is really interested in proteases. And viral proteases are very interesting, especially in positive strand viruses, which work by this polyprotein strategy, as you know. And so every protein is made in stoichiometric amounts. So really all they have for regulation is a sort of the systematic proteolytic digestion. What I want to tell you about these proteases, so for example, the embedded proteases in picornaviruses 2A and 3C, the embedded protease in dengue virus and other flaviviruses, NS2B3, is they have a lot of different kinds of cleavages. They can cleave themselves out of the polyprotein, and they can do that either intramolecularly, only in the polyprotein from which they're made. They can do that intermolecularly, working on other polyproteins, and they all have about 20 to 50 host proteins that they cleave. So what is it that we should be optimizing if we want to make an inhibitor of a protease? Because it's not like thrombin, thrombin or something that has only a few substrate, it has about 40. So how do we know which is the correlate of inhibiting virus growth and which we should be targeting? So I'm going to dissect now for you some of the cleavages in picornaviruses and flaviviruses. Okay, so here's a diagram of picornavirus genome. Like I said, it's made as a polyprotein with a whole bunch of, these are the capsid proteins. There are two proteases called 2A and 3C. 2A makes exactly one cut in the polyprotein between itself and capsid protein VP1. When this cut isn't made, you accumulate a precursor naturally, and we don't know quite what that precursor is, whether it involves more of the proteins or not, when this cleavage is not made. 3C makes a whole lot of cleavages in the protein. These are made as its so-called precursor 3CD in the capsid proteins, but the 3C active site is responsible for all those cleavages. And the question is, we know for 2A that it makes only this cleavage in the polyprotein, but it cleaves about 20 host factors too. 3C makes all these cleavages, and it cleaves about 50 host factors. So which are important, and how do we study it? Okay, so what we wanted to ask was, for these cleavages in the polyprotein, does 2A cleave this bond intermolecularly or intramolecularly? Ditto for 3ABC, is this an intramolecular or intermolecular reaction? And you might ask, why do you care? And I'll show you that later. But first, let's talk about how do you even study an intramolecularly cleaving protease? I mean, you can't really purify it because it'll cleave itself and you'll end up with VP1 and 2A, for example. So what we do is we make them in rabbit reticulocyte lysates and catch a time point at which the labeled protein still remains partially as a precursor. Then we add a cold methionine chase and watch that labeled precursor get degraded and measure the kinetics of that. So I'll show you an experiment like that. So here's a question. If we express VP1, 2A in a rabbit reticulocyte lysate, Here's VP12A at 0 time points of chase. You can see under the 20 minutes of synthesis that preceded that, you still see precursor and you see some products too. But now we're going to ask, at what rate is the precursor cleaved by the activity of 2A that's within it? And if the cleavage is intermolecular, as you dilute this extract, the rate should decrease because it's a two-body reaction. If it's intramolecular, it should not change with dilution. And that's in fact what you see with poliovirus VP12A. In this case, you see no change in the cleavage rate with dilution, arguing that that cleavage is intramolecular. There's another way to study this too, which is here's an uncleavable precursor because we mutated the active site in 2A. What happens when you mix in a cold functional VP12A? Does it get cleaved? And the answer is yes, a little bit. So some happens intermolecularly or in trans. What happens when you just let it cleave by itself? Does a precursor disappear? Yes, a lot faster. So that's plotted here. Here's the uncleavable one. Here's one in which you can have either inter or intramolecular cleavage. And this is the one in which you have inter alone. So you can see while some of this cleavage can be intermolecular, most of it's intramolecular, consistent with the dilution independence. So why do we care about that? Let me show you this here. So let's talk about an untreated, we're talking about just an untreated cell that's making virus. The polyprotein is drawn here kind of like a ball of yarn, you know, a folded polyprotein. And 2A is going to cut it, right? And so it's either going to cut it intramolecularly, as we've just observed, in which only this 2A can cleave it, or they're going to be able to cleave each other. So under this circumstance, when you inhibit 2A protease, that's going to be inhibited whether it cleaves intramolecularly or intermolecularly. And under both circumstances, they'll accumulate this VP12A product. So the inhibition could either come from the failure of the protease to work at all, or maybe this product is a little bit inhibitory. However, what happens when it gets interesting when you have a drug-resistant variant that shows up in the cell? This can still cleave itself if it's intramolecular. Those can't. This one can't cleave those because it's intramolecular. So the precursor will persist, unlike if the cleavage is all intermolecular. Therefore, if these accumulated precursors are dominant inhibitors, as is often the case with precursors in positive strand RNA viruses, then every virus in the cell will die. Therefore, that's why we're pretty interested that this cleavage is mostly intramolecular. What about those viruses that are the subject of the AViD, interest in the AViD program? You can see these experiments again. Here's the cleavage of VP12A of enterovirus A71, and you can see that it's almost exclusively intramolecular. And that's also true for the protease of AViDD68. So it's exclusively intramolecular. And does that mean, and so we suggest that that might mean that the defective genomes would be dominant over the wild type genomes because a precursor would accumulate that would be toxic. Is that the case? Well, here's an experiment in which here's a poliovirus genome, and wild type poliovirus is mixed with a variety of dead viruses. This is one with a 3' untranslated region that's defective, and you can see that, you know, it reaches some yield. Here's with a genome that has a mutation in the 2A protease. You see a drop in a log of, even in this first cycle of infection, of yield of the wild type virus, just because this defective genome is around. And here's one that's cleaved at the VP12, that's defective at the VP12A cleavage junction. So it's another way to block cleavage. And that also has a dominant effect. Okay, so let me show you enterovirus A71. Here we don't have any of those other mutations because the genetics isn't so well developed, but here's the yield in the presence of a lot of tRNA, and here's the yield in the presence of a defective 2A genome, and you can see a lot of dominance, and that's true also for AViDD68. So we think that targeting intramolecular cleavages is a good way to make protease inhibitors that will not only inhibit the virus, but will suppress the outgrowth of drug resistance due to the accumulation of toxic precursors. But what do existing antivirals do? Do they do that, or do they do something else? So as you know, there's some really nice papers from Jun Wang's lab in which they've shown that telaprevir is a very effective inhibitor of enterovirus D68 growth. It also inhibits the protease of AViDD68. And so is that a direct correlate? What is the direct correlate of inhibiting viral growth? What substrate of 2A protease is the cleavage of which substrate is inhibited? So here we're looking at an experiment like I told you before. Here's the accumulation of VP12A from AViDD68. You can see just a little bit of precursor there. As you increase the amount of telaprevir, in the experiment, you see a little bit of inhibition of that cleavage. And you see it here, that it's a very little inhibition of it, although it is a little bit inhibited, but the EC50 is greater than 1 millimole and 1 micromolar. Okay, now here's one of those host proteins I told you about that gets cleaved by viral proteases. This is an infected cell extract, and this is EIF4G, a translation factor that, as a lot of you know, is cleaved by various viral proteases during infection because it blocks, then host translation is blocked. In the case of enteroviruses, a little fragment of EIF4G also helps with viral translation, so it's very important for viral growth. And you can see that in the absence of telaprevir, you get a lot of cleavage of EIF4G. This is in the rabbit reticulocyte extract, so this was detected by immunoblotting. You see a lot of cleavage of EIF4G, and that starts to be inhibited by telaprevir with an EC50 of, that's a lot lower than that of the polyprotein. So here's a question. So telaprevir is a much more effective inhibitor of cleavage of this host protein than it is of the viral polyprotein. So why does it inhibit viral growth? There are kind of two potential answers to that question. One, its real substrate is this host protein, and that's fine. That's a fine way to inhibit a virus. It's just kind of surprising that it wouldn't be the viral protein. The other explanation is, well, maybe this cleavage here this inhibition here is sufficient to inhibit the virus, because if it's generating a toxic precursor, maybe you don't need to have 100% cleavage if you're not just looking at loss of function, but gain of a strong inhibitor. So to be continued. Okay, so what we've learned is that the enterovirus 2A proteases all cleave almost preferentially intramolecularly, and that telaprevir can inhibit that cleavage, but is more effective at inhibiting EIF4G cleavage. What would happen if we optimized our inhibitors to really go after the VP12A cleavage that's in the viral polyprotein? I guess my suggestion is let's assay that as well. Okay, what about 3ABC? Well, we did the same kind of experiment to ask if those cleavages were intra or intermolecular. And to make a long story short, let's just look at the dilution sensitivity. Here's the little bit of the polyprotein we tested, that is poliovirus 3ABC, and you can see that its cleavage is dilution sensitive. That is, it's very consistent with being an intermolecular reaction. But in the case of AViDD68 3ABC, which is one of the targets in the ASAP program, you can see that its cleavage is intramolecular, therefore making it a kind of a different kind of substrate to go after. And we're pretty excited that maybe there'll be some dominance effects associated with that. What is the effect of ropintervir, an inhibitor of 3C, on these cleavages? Well, here it is inhibiting the intermolecular cleavage of poliovirus 3ABC, and you see that it inhibits, okay. It has an EC50 of about 0.2 micromolar. And here it is inhibiting the intramolecular cleavage of AViDD68 3ABC. It also inhibits pretty well, but about 10-fold less well than this. Does this correlate with the EC50 for the virus? Well, the intermolecular reaction of poliovirus is inhibited at pretty low concentration. And you can see that that corresponds to a viral inhibition of 22 nanomolar. This is 10-fold worse at inhibiting the biochemical reaction, but it's got a better EC50 for the virus. So is that these assays were done by different people in different labs, but we're wondering if maybe what we should be optimizing is the intramolecular cleavage, is to choose variant proteases like this that have intramolecular cleavages and optimize that reaction. I'm going to skip this because it's published data from dengue virus that shows that precursors can be dominant. And I'm going to talk to you about quasispecies again. So we know that we can dominantly inhibit the growth of viruses within infected cells in the first round of infection. That's some of the stuff I was showing you with co-transfection. What happens in cells? What happens during viral spread? You know, we really don't know how viral spread works in a lot of systems. So we wanted to investigate how long does a quasi-species effect persist? And if you look at, this is an organoid, a colloid derived from human colon stem cells. And we're going to, this is the actin, it's got an apical out topology. And you can see here's the actin, the cortical actin layer here. So if you infect with enterovirus A71, what you see is you can see some infected cells. The green is the double-stranded RNA from the virus. So you see this cell is infected. So Jasmine Moshiri, a grad student in my lab, it was her project to watch the virus spread. And that was kind of frustrating because it didn't spread. We never saw cell-to-cell spread, even though we saw a lot of increase in viral titer in the medium. So she was pretty frustrated, like what was her project? And then we noticed that it didn't spread within the organoids because the cells got extruded. In fact, infected cells are specifically extruded, and then they end up free in the medium. If you ask what happens to those cells after they get extruded, they stay, about half of them stay viable for about 20 hours, and they're viable in bile, and they're viable in fecal slurries. So the point is that the quasi-species can develop in the first infected cells, but in some cases, viruses, as we know, spread en bloc. That means altogether. So these quasi-species effects can be continued throughout for a long time during infection. So basically, we're hoping to, within our consortium and together with all of you guys, to figure out how we can make good drugs, but also to have them persist in their utility. Of course, we all think that we could combine our resources to do combination therapies, but some attention to the genetics before targeting might be a good idea, like thinking about fitness costs as we design the substrate envelopes based on, you know, sort of the high throughput, high intensity genetics that are now capable. We also like to choose our targets based on the potential for making the drug-bound proteins be dominant inhibitors. And so that's something we will be exploring, especially in the intramolecular cleaving protease domain. And that's what I have to say. So thank you. And I look forward to talking to more of you about your explorations in antiviral development as well.